and it's because it's a seminar that the speaker was selected by students. So we have a student committee, which Allison is the chair of, and they met, discussed many candidates, voted, and so we have, I'm not going to say who it is because that's her job. So we have our first student selected speaker here, and so this is a really exciting day for this seminar series. So without anything else, Allison.
Thank you, Mike, and good morning to everyone. It is my great pleasure to introduce our speaker for today's Pioneers in Biomedical Research seminar, Dr. Clare Waterman. Dr. Waterman's contributions to cytoskeletal cell biology and the methods used to study these systems are profound. Her research has been featured in more than 100 articles in top journals including Science, Nature, Nature Cell Biology, Cell, the Journal of Cell Biology, and many more. While a postdoc in Ted Salmon's lab at UNC, she helped to discover high-resolution microscopy technique called fluorescent speckle microscopy that allows for submicron visualization of cytoskeletal dynamics. She continued that exciting work in her time as an assistant and then turned tenured associate professor at Scripps by teaming up with a computer engineer to develop image analysis software to map cytoskeletal assembly and disassembly processes, as well as the motion in migrating cells. She also used these techniques to introduce data supporting the concept of cytoskeletal crosstalk, that is, the direct interactions between cytoskeletal systems. And after her time at Scripps, she was recruited to the National Heart, Lung, and Blood Institute of the NIH, where she serves as the director for Cell Biology and Physiology Center and the chief of the Laboratory of Cell and Tissue Morphodynamics. At the NIH, she developed high-resolution traction force microscopy, which allows the quantitation of the direction and magnitude of cell-exerted traction stresses on an extracellular matrix at submicron spatial resolution. She used this technology to show how actin mediates the tugging of the cell extracellular matrix and how this is necessary for guiding cell movement. Dr. Waterman has been the recipient of numerous awards, grants, and prestigious lectureships. Notable examples include the NIH Distinguished Investigator Award, NIH Director's Challenge Award, NIH Award of Merit, the Arthur S. Fleming Award for Public Service, and Frank R. Lilly Research Innovation Award. She is a member of several professional societies and has served on the editorial board for multiple publications, including Current Biology, the Journal of Cell Biology, Molecular Biology of the Cell, and the Journal of Microscopy. On top of such a substantial career, Dr. Waterman has also excelled as a mentor with many of her former trainees, obtaining tenured or tenure-track positions in an ever-increasingly competitive environment. On that note, I would like to share with you that Dr. Waterman is the first student panel-nominated speaker to be invited to the Pioneers in Biomedical Research seminar series, as Mike mentioned. It is an honor to have her with us today, and I would like to personally thank her for agreeing to come speak with us about her exciting research endeavors. Without further ado, Dr. Waterman will be sharing her work with us in her talk titled Integrating Actin and Adhesion Dynamics in Cell Migration.
How do I do this? Janet Jackson. Oof, that's weird. It doesn't really...
Upside down.
Upside down? I think it's right side up. It goes...
The curvature actually goes over your ears, so it is...
Oh, this way. Okay, it's like
Yes,
the
yes.
backwards glasses, I see. Like that. Okay, all right, I got it. But it still is way up. Okay, all right, all right. Okay. All right, well, thanks for that really nice introduction. My mother always says, well, they must give an awful lot of awards in your field. No, it's a pleasure to be here, and it's an honor to be asked by students. And I didn't know about this campus when I got invited, and she said, she invited me to somewhere in Roanoke, Virginia, and I thought it was near the beach. But it's exciting to see this place. It's really impressive, and it's amazing what they're building up here, a new medical school, a whole new neuroscience institute, and it's good scouting for my postdocs that are out there looking for jobs. And what I'm going to tell you about today, I decided to talk about something that's completely new and Colleen actually is on the job market. I don't know whether she's come through here yet, but this work is all hers that she's been working on for the past several years in my lab, and it's sort of a new direction, getting into more, probably I would say a little more clinically, physiologically relevant than the basic studies that I've been doing throughout my career on the cytoskeleton. And we sort of just fell into that. It was not like we were going that way, but I'll tell you how we got there, okay? So what I'm going to tell you about is this actin nucleator that makes actin fibers that protect the nucleus, and when cells are migrating in 3D microenvironments, it really protects the nucleus and keeps it from getting damaged. So it's a weird function for an actin system. So my lab is interested in the fundamental mechanisms by which cells migrate. And do I have just a pointer here, like a laser pointer? I don't want, this is gonna like, yeah, okay, I don't want to be switching my slides without knowing it. I hate when that happens. Okay, so what you see here, this is a developing zebrafish, and the endothelial cells are lit up with GFP. The fish is like upside down, so its dorsal fin would be up here, its heart would be up here, and its tail would be down here. And at a certain point in development, this is about, I don't know, like 13 days or I don't know what the timing is. But you get these cells receive a signal to migrate through. These are developing developing muscle segments, and you have these endothelial cells that migrate in a very directed fashion in a specific direction at a certain time in development. What they do is they migrate around and they touch each other and they make these loops so that now blood can flow around each one of these muscle segments and supply it blood. So what you see there is it's a very tight timing of the initiation, and it's a very, very highly regulated directional path that the cells are taking. So this is an example of directed cell migration that occurs during everyday development of an embryo. Now, once you're an adult, you also rely on cell migration for the maintenance of your health, both the remodeling of your vasculature after injury. This is an example of immune cells migrating. This is also a zebrafish. Now this is it's turned over, so the dorsal fin is down here, and what's happened is the dorsal fin has been poked with a needle, so you've got a wound, and the blood is flowing through this dorsal aorta. The cells in this blood feel diffusible factors or smell diffusible factors that are released. The white blood cells get their integrins activated on their surface. They grab onto the endothelial lining. They squeeze their way through the endothelium, migrate through this epidermal tissue, find this wound, eat the bugs that are in there, eat the dead cells, start activating the adaptive immune system, go backing, presenting antigens in there and whatever. So, you can't heal these wounds, you can't remodel your vasculature without cells being told when and where to migrate in a timed and directional fashion. And if you mess this process up, if you get deregulation of this process, there's a multiple of a ton of diseases that are resulting from misregulation of cell migration during development, as well as in your immune system, chronic infections, multiple sclerosis, vascular diseases such as excess vascularization of the retina. This is what my mother has, and I'll probably get. And sort of the classic example of deregulation of cell migration is cancer metastasis. This is a movie from Eric Sahai when he was in the Condillason lab. This is a melanoma cell tumor. So the melanoma cells are green. That's been implanted in the mammary fat pad. You can see the collagen fibrils that are, just supporting that tissue in purple. And you see cells that are crawling out of the tumors through this complex collagen matrix that are getting into the blood and the lymph system and going on to spread the cancer throughout your body. So you really want to stop it in this situation, you want to stop it in this situation, and in other situations, you might want to start it. So we need to be able to control this process in order to control these really nasty diseases. So when you get down to the cellular level, sort of the classic view of cell migration is that cells crawl across the substrate by protruding the leading edge in the direction of migration. That protrusion has to adhere. Cells have to generate force between that front adhered protrusion and the middle of the back of the cell to pull the cell along, and then they have to let go in the back. So the whole process is regulated in time and space just to initiate it and have it go in the right direction. But then even at the subcellular level, it's about spatial and temporal control of these subcellular processes. And this cartoon, this is an actual cell doing what that cartoon was drawn after, and the markers, you see the nucleus in blue with a histone, that's actually a cardinal, a far red tag. The actin cytoskeleton is in green, and the adhesions are in red, and you can see protrusion happening at the leading edge in concert with the formation of these tiny little adhesions. These adhesions undergo a maturation process that's due to the contraction across the cell. They mature, hold the cell down, and then these ones stay strong in the front, and these ones in the back get weak, and that pulls the cell along in a certain direction. So it's this highly spatially and temporally regulated process involving the cytoskeleton, its proper assembly and formation into structures that mediate this highly regulated process. So if you look at the actin cytoskeleton, these are migrating fibroblasts. The protrusion is happening up here at the leading edge through this dense assembly of actin called a lamellipodium. But there's all these other structures of actin of actin filaments that are participating in this process. So you see this sort of fine cortical meshwork. This is maintaining the sort of structural integrity of the cellular cortex. You see these stress fibers that are, one of these is stuck in an adhesion, and it can go back all the way and meet with an adhesion on the other side of the cell. Then you've got these arcs that are sort of across the top of the cell that hold the top of the cell down and maintain the shape. And then you've got a bunch of stress fibers underneath the nucleus. One of the question that sort of Colleen was focused on is how are these diverse actin structures made, and what are they used for specifically during cell migration? And so how do you make actin? You make actin. Actin is made out of monomers of actin that are at high concentration, 100 micromolar in a cell, super high concentration, but they don't assemble into a filament. The formation of this nucleus, that three monomers come together in a specific geometry that allows assembly of more actin monomers onto the end of that, is a really rate-limiting process. If you look at these arrows, these are equilibria arrows. It's like really, really slow to go in this direction. So this is a highly regulated. So there's proteins that control where and when this happens at a cell so that you can make structures where and when you want to make them in a cell. So these proteins are called nucleators, and they basically increase the forward arrow by making sort of a substructure that favors the formation of this trimer that then can go on and have actin monomers add on to that easily. Okay, so these are called actin filament nucleators. Probably the most famous one is the ARP2/3 complex that makes the lamellipodia in the leading edge of the cell. But there's other classes that have been more discovered a little more recently and are less well understood. So the ARP2/3 complex binds to the side of an existing actin filament and nucleates a new filament off that side. So it makes a branched network that's sort of like a gel if you look at the macroscopic properties of it. It's like an expanding gel. Formins help form a trimer, and then they stay associated with the actin filament, and these arms that stick out add actin monomers underneath themselves. They stay associated with the end of that filament, and they can regulate how fast that filament grows. But they make linear filaments, usually they contribute to the formation of actin bundles. We saw a whole bunch of bundles in that migrating cell. And then even more recently and less understood is the spire family, which also can make linear filaments, but really it's wide open. We don't have much knowledge of either the biochemistry or the cell biology of these proteins. So this talk is going to be focused on a member of this Formin family. So these are the three, the ARP23 makes the branch filaments, and the Formin and spire makes straight filaments, and Formins, in addition to nucleating them, can control their elongation rates. So the whole field of cell migration has been totally obsessed. Well, we discovered this ARP23 complex when I was a graduate student, and the whole field has been obsessed with how this ARP23 complex makes the lamellipodia at the leading edge of the cell, because this is the protrusion. This is the exciting part of cell migration. It's moving the cell forward, you know? We're all obsessed with the front of the cell, and so this is in excruciating detail, the biochemical regulation of the formation of these filaments at the leading edge. Hundreds of papers published on this. But if you look at the migrating cell, we're all obsessed with this tiny little thing, and nobody's studying what the hell is going on in the whole rest of the cell. But, nobody likes the back. Everyone likes the front. So, I went around talking about the front for years, and graduate students in the audience would always say, so what regulates the back? And I'm like, well, nobody really studies it. It's like embarrassing, because everybody loves that front. Everybody wants to know how it moves out. So the ARP23 makes this sort of dense gel at the edge that drives the edge forward. But then there's all these meshworks and bundles back here that we don't know who makes them or what the function of them are. Okay, so there's filopodia that can be at the leading edge of migrating growth cones, sort of feeling out the territory. There's these transverse arcs that sort of maintain the shape of that lamellum and keep it flat. So these are on the top of the cell, and they sort of hold it down. There's these dorsal stress fibers. They terminate in a focal adhesion, and they can generate traction force on the ECM. There's actin structures on top of the nucleus called tan lines, transverse actin, I don't know what it stands for, but there are these stress fibers that go over the top of the cell, and there's bundles in the back. And we don't know much about how any of these things are made. And they're linear structures, so we know it's probably not ARP23, and it's probably going to be Formins. So Formins are this ARP23. There's only one of. There's only there's not isoforms. There aren't related family members. There's one ARP23 complex. Formins are a much more complicated class of actin nucleating proteins. These are the ones that are in the human genome, and there's lots of spliceoforms even of them. Okay, so it's a large family of actin nucleators, and what Colleen is interested in is who makes what. So some of them we know. We know some of the dias make filopodia or these transverse arcs. There's an INF, inverted formin that makes actin filaments that regulate mitochondrial division. There's DAM1 that makes this sort of veil-like network in between all the stress fibers. But we don't really know, is it like kinesin motors? Is there one for every structure? And if there's one for every structure, which formins are making which structures, and how are they regulated? So this is sort of Colleen's, she came out of Dave Kovar's lab studying the biochemistry of these formin proteins, and then came to my lab to figure out what these proteins do in a cell. So the hypothesis is that different formins can build different structures to drive distinct aspects of cell motility. So when Colleen came to the lab, we had just done a proteomic analysis of the focal adhesion. So Jean Cheng worked out a method to isolate focal adhesions, threw them in John Yates's mass spec, and we got out 900 proteins. And Colleen looked through that list, and she said, oh, there's two formins in there. So the formin I'm going to tell you about today is this FMN2. So it was found in our focal adhesion proteome, meaning that it might be somehow associated with the link between actin and adhesion. What's known about FMN2? Not much. So there's like 2 papers on the biochemistry of it. It has the sort of canonical thing that defines a formin is this FH1, FH2 domain. That forms that, you saw that lock washer that goes on the end of the formin in the cartoon. That's what forms that lock washer. Then these parts are divergent between the family members. This is just data showing that this is an actin polymerization assay. This black line is actin by itself, and then when you add formin, it increases the rates of actin polymerization. So it gets you over that energy barrier for the nucleation of actin filaments, so it's an actin nucleator. And then there's some structure that has been done on its interaction with a spire protein, but there really isn't much known about it. So it interacts with this other actin nucleator. There's a lot more that's sort of out there about it from the literature from genetics. So it's expressed, highly expressed in the developing nervous system, also in oocytes. It was discovered in Drosophila oocytes, basically. Its deletion, probably due to the nervous system expression, results in mental retardation. It's also associated with cancers. It's targeted by a microRNA in certain metastatic diseases. Mutations are associated with breast cancer, colorectal cancer, also the sort of oocyte connection. It's connected with infertility. And it's overexpressed in leukemias. So this is a pretty interesting actin nucleator. You're not just dead. You know, like ARP23, you're freaking dead. You don't have ARP23, you don't get past the 1-cell stage. So the fact that there's many formins means that they end up contributing to diseases in humans because they can, take over each other's jobs a little bit. So what's known cell biologically, the only thing that's known cell biologically is about what it does in oocytes, and there's a couple of papers on this. This is basically what it does is it positions the mitotic spindle near the periphery in an oocyte, and this has been seen both in invertebrate oocytes as well as mouse oocytes. So it makes this actin meshwork in the egg that helps position the mitotic spindle. But we found it in a focal adhesion proteome that was made from human fibroblasts. So what is it doing in the adhesion proteome, and is it doing anything in cell migration? So Colleen started working on it. We looked at the localization of this formin, and right off the bat, that was weird, okay? So, we're all obsessed with the leading edge, and then we saw this formin, we're like, oh my God, it's like right under the nucleus. It almost looks like it's in the nucleus, but this is a confocal section on the bottom of the cell, it's underneath the nucleus, and it's associated, it's not totally perfectly co-localized with this actin and these adhesions under the nucleus, but it's associated with this sort of subnuclear actin and adhesions. And this we looked at in multiple cell types, so endothelial cells, fibroblasts, and adenocarcinoma cells. When you look at it, you make a GFP-tagged FMN2 and express it in a migrating fibroblast, migrating on fibronectin. It forms this sort of cage that follows around the back of the nucleus. I've never seen anything like this in my life. It's fibrillar, but I'll show you in a second that it's not actin-dependent fibrils necessarily. So as a cell migrates, it sort of makes this cup around the backside of the nucleus and follows the nucleus around. Sort of look like a little cradling it almost, So is it associated directly with the actin cytoskeleton? Well, you can disrupt the actin filaments by dumping latrunculin on. And what happens to it is it collapses into these little round rings. And I'm like, oh, that looks like vesicles. I wonder if it's, and those fibrils, I don't know what they were. I thought they might be intermediate filaments. It doesn't localize to intermediate filaments. It doesn't localize to any, RAB compartment of the endocytic recycling compartment or whatever, but it is triton extractable. So if its extension into these fibrils is dependent on the cytoskeleton. When you get rid of the cytoskeleton, it collapses into membrane vesicles that are extracted by triton. So it's associated with some weird membrane compartment that follows around the nucleus. My pet hunch is that it might be some kind of extension of the nuclear envelope, but I don't have any evidence for that. I'm just guessing. So what are these? Okay, so it's associated with the subnuclear actin and adhesion population that nobody's ever paid any attention to. So what are these subnuclear actin and adhesions? Are they anything special? Well, we first just analyzed the composition of them. So, stress fibers have alpha-actinin, tropomyosin, VASP, myosin 2 on them. So we looked at all of these markers, and really so here what you're looking at is like the leading edge stress fibers versus the underneath the nucleus stress fibers. Leading edge stress fibers have VASP, these ones don't. They don't have any alpha-actinin on them. They don't have any myosin 2A on them, and they do have a lot of myosin 2B. So this is sort of just, stress fiber aficionado stuff. Are they contractile? Well, to see if they're contractile, you want to know, are they capable of generating any force? So Colleen did a photo bleaching experiment where she bleached 3 stripes on them and then monitored the position of those stripe marks over time. This is a chymograph, and what you can see is that the stripe marks move towards the center. The center stays constant, but it recovers its fluorescence. So that means that actin is going in towards the middle from both ends. So they're contractile. So they're contractile myosin 2B containing stress fibers under the nucleus. These stress fibers under the nucleus actually sort of impinge on the nuclear envelope. So this is a histone, and then we're looking at myosin 2B, and you can actually see they sort of deform the bottom of the nucleus. So they're really in close contact. There's these contractile stress fibers under the nucleus that are in close contact with the nuclear envelope. When we watch the adhesions that are under the nucleus, which I've never looked at in my life, I've looked at adhesions for 15 years. But, the camera in a high resolution, we're always, 100x 1.4 NA lens, you don't see the back of the cell. So if you're in the front of the cell, you don't even see what's going on in the back of the cell. So it's like embarrassing, you spend 15 years looking at one part of the cell, it's pinheaded. So here's this is a big, beautiful fibroblast sending out protrusions. There's this subpopulation. These adhesions that are out here, they were all born at a leading edge. Okay, so they're all born when a lamellipodia gets sort of rolled out like a carpet. These new adhesions form, and then they mature into these big adhesions. These adhesions under the nucleus have nothing to do with the leading edge. They appear out of nowhere, and they sort of treadmill along with the nucleus. So they're born by a different mechanism. They have a different lifetime and dynamics as typical leading edge adhesions. So this is like a subset of actin adhesive structure that hasn't really been described, and it's not like stuff going on at the leading edge. These adhesions don't remodel the extracellular matrix. So this is a fluorescent fibronectin, and you can see fibronectin fibrils forming underneath adhesions that are at the leading edge. If you look under the ones that are under the nucleus, this is a time lapse. It's not really moving that fibronectin around. So they aren't. That's what fibroblasts sort of do for a living is repair extracellular matrix. These aren't involved in that process. It doesn't appear. So, okay, is this FMN2, this Formin that looks like it's near there, is it making these funky stress fibers and adhesions? Yes, okay, so you get rid of that Formin by knocking it down by siRNA, and now you have no adhesions under the nucleus, and you have none of those stress fibers in the middle of the cell at all. And you can quantify this just by counting. There's no subnuclear adhesions when you knock it down, no ventral actin bundle. So this is done by TURF, so we're only looking at the actin on the bottom of the cell. We can do a 3D reconstruction by confocal. These stress fibers are specific to the bottom of the cell, so they aren't tan lines. What is it doing, what is it doing under there? It's sort of following the nucleus around. Does it have anything to do with how the nucleus is positioned? So this is, I hate this slide. I wish I had a movie, but she didn't give me a movie. So this is the centroid of the cell and the centroid of the nucleus. And if you track, as a cell migrates around, the nucleus tracks with the centroid of the cell. And in an FMN2 knockdown cell, they aren't perfectly correlated. So the nucleus sort of goes over here while the cell is going over there. So the nucleus is sort of wandering around a little bit inside the cell. If you're familiar with cell polarity studies where you make a wound at the edge of a cell and the cells start migrating into the wound and the MTOC orients in front of the nucleus, that process is messed up a little bit too. So you got a little bit nuclear positional defects. This is just showing that it's not just in an X and Y. So we do simultaneous epifluorescence, which has a thick depth of focus, and turf, which has a really thin one. And so where those overlap, you see yellow. This is the knockdown cell. The nucleus is actually sort of floating up and down as well as drifting around in X and Y. The other thing we noticed is not only do you have this sort of bouncing around, so this is the time course of the position of the nucleus over time, you have these big, nasty lobules on them. These are primary fibroblasts, less than 10 passages. The nuclei tend to be beautiful oblate nuclei, not like in a cancer cell. We noticed that these FMN2 knockdown cells had increased lobularity, and the sort of axis of the nucleus and the axis of the cell, that correlation was decreased. But you got these big sort of bulgy things sticking out of the nucleus. The nucleus is bouncing around a little bit in the cell, but really the cells sort of migrated fine. They could migrate pretty well, no problem. Maybe a little polarity defect, but they migrated pretty well. They divide fine. So is this thing do nothing? We're sort of bummed out at this point. But I was freaked out by this. I'm like, that is effed up looking, These are primary fibroblasts, and this is a bulgy nucleus that looks like a cancer cell. I don't like that, Let's figure out what's going on, Colleen. I'm like, who cares if the nucleus bubbles and drifts around a little bit during migration? Is this like protein doing nothing? But that bubbled nucleus looks really messed up to me. Okay, so, cancer cells have bubbly nuclei. Cancer cells crawl around in complex 3D environments in order to get out of tumors. They got to cross through a basement membrane. They have to crawl through, like in the case of vascular cells, between these muscle segments in a fish wall. Cells have to be challenged a little bit more in their environment. So what about if we put these cells lacking FMN2 in a more physiological environment? So this is the same fibroblasts that make those nice flat pancakes that are shaped like ice cream cones on a 2D collagen gel. When you stick them in a 3D collagen network, now they start looking a little bit more like a tip cell of an endothelial chain. They take on a completely different morphology, and they're doing more like what we want to try and stop, which is a cancer cell getting out of a tumor. So now we look with a confocal microscope and look at the localization of this FMN2 in a fibroblast crawling in a 3D culture, and the same sort of localization. It's around the nucleus, following sort of the back of the nucleus, and if you do a 3D reconstruction, it's like a cup that's around the back of the nucleus, and even when the cell changes direction and the nucleus starts going in the other direction, it stays around that nucleus and follows it, sort of like, it just looks like it's like supporting it as it goes. These cells in 3D actually do have stress fibers that surround the nucleus, and if you knock down FMN2, you lose those stress fibers. So similar to the 2D phenotype, you also lose the adhesions. There are a couple adhesions around the nucleus. You have no effect. You knock down FMN2, you still have the leading edge adhesions. Hard to see adhesions in 3D, but you lose these foci of vinculin that mark the adhesion. So FMN2 is creating sort of an actin adhesion system that's surrounding and following the nucleus as the cell migrates through a 3D collagen network. But when Colleen was doing these experiments, she's trying to look at these cells in 3D, and they're all just dying. Like, she couldn't keep cells alive for more than a few days. And we were like, that's pretty weird. So is this thing required for survival in a sort of invasive migration situation where you got to crawl through a dense 3D matrix? And so we subjected them to an invasion assay. So this is sort of like classic invasion assay where you make a matrigel or, you know, a collagen network that you seed cells in, and you put, and that's on a permeable membrane that has pores in it that the cells can migrate through, and you put some growth factor down here and the cells migrate through. And what we found is that we just weren't getting any, we had the FMN2 knockdown cells marked with a GFP. We didn't get anybody coming through there. So they just weren't making it through. We did a Western blot on the top and the bottom. We weren't, we didn't have a reduced population, I mean, an increased population of cells on the top, you know, so I think they were just croaking when they were trying to get through. So we're saying, okay, so what is different about invasive migration? Well, the cells have to squeeze through things, and in that transwell assay in particular, you know, you can get ones that have like 8-micron pores or 12-micron pores, so it's pretty small, and the nucleus of a fibroblast is about, it's about 12 microns by about 15 microns. They're oblong, but they're pretty big, so maybe it was having trouble, maybe you know, this cage was somehow protecting the nucleus as it tried to get through this, these small pores. So we collaborated, actually Hawa, she's a new postdoc in my lab that is from Matthew Piel's lab at Curie, and she had made these PDMS microchannels for her PhD work in Matthew's lab, and they were perfect. They're these little channels that are about 7 microns in the fat part, and then they have really narrow constrictions that you can control the size of. So in this case, it's a 7-micron channel. You seed cells over here. You don't even really have to put a gradient of growth factor. They'll just go in there and crawl across. So these are movies of the cell that has a nuclear localization signal to mark the nucleus. And when you watch, the top ones are a control cell, the bottom are the knockdown cell. And what you see is the control cell, it gets through there fine, it squeezes the nucleus through that tiny little space, which is sort of amazing. This guy, a lot of times they croak before they even get to the tight constriction, and their nucleus just explodes. Okay, the NLS, actually you see that this one, it does release a little NLS when it's squeezing through. So there is a little compromising of this, either the import, the export, or there's tearing of the nuclear envelope that's getting repaired. So you do have a little bit of leakiness of the NLS even in a control cell, but this cell just can't even make it through. It just explodes. The nucleus explodes and the cell dies. So this is the fat part. A lot of them die even in the fat part, and pretty much all of them die going through the constriction. You have a little bit of increase in death for the controls when they try and get through the constriction, but most of them make it through. I mean, it's actually amazing that you can squish the nucleus and squeeze it through a 3-micron radius hole.
You say that these are normal fibroblasts.
Yes.
And in cancer cells, FMN2 is overexpressed?
It is overexpressed in some, it's mutated in others.
Mutated to make it constitutively active?
I don't know. I don't know what, you know, it's just like, you know, in people's chip studies and people, you know what I mean? There's nothing really specific known about it. Okay, so you know, when we were just starting to think about like the nucleus getting smushed and smushing these cells through channels, a paper came out from Foiani lab in Cell showing that you can actually damage the DNA just by smushing a cell. And then work that was going on that is just going to come out. It's going to come out in Science like 2 weeks from now. And Hawa knew about it because she was in Matthew's lab. She's like, I'm getting really uncomfortable. You guys are starting to work on something that was totally going on in my lab before I left that I wasn't working on. So I called up Matthew, told him what we were doing, and he was like, oh, that's totally awesome. Let's talk about this. He told me all his results. And basically what he found is that nuclear envelope rupture is going on a lot. And when the nuclear envelope ruptures, it usually repairs. But if it doesn't repair, like he has like a, I don't know what it is. It's like it's it's one of those membrane, like it's not a snare, it's maybe a snare or one of those membrane trafficking things that's involved in fission and fusion of membranes, that if you mess that thing up, the nucleus can't repair as fast. And what happens is the DNases from the cytoplasm get into the nucleus and cause double-stranded breaks. So compromising, mushing on a nucleus or tearing the nuclear envelope can result in DNA damage. And the way that you assay DNA damage, this is DNA damage that's induced by gamma irradiation. When you have double-stranded breaks, you set up a DNA repair complex that can, I don't know much about this. It's not my area of expertise. We're actually collaborating with Andre Nussensweig, who's in the NCI, and he's a big DNA damage expert. So you set up this repair complex, you get this histone that gets phosphorylated, gamma H2AX gets phosphorylated, and this DNA repair protein 53BP1, they form foci right at the site of damage, and they repair this damage. So you get a lot of them when you subject the cell to gamma irradiation, and the sort of upstream regulators of inducing this repair complex that the DNA repair people worry about are these kinases, ATM and ATR, and there's two different pathways that are induced by different stressors, okay? I don't know much about it, but they're like, totally worry about this. Okay, so we can assay it, and so what we did is we stick cells through transwells. We look for the ones that actually make it through and are green, and we know that FMN2 is knocked down. So we take the cells, put them through transwells, collect them, replate them, stain them from gamma H2AX 53BP1, and look for the ones that are transfected. You can tell right away by looking at the actin. It doesn't have its subnuclear adhesions and stress fibers, and it's got gamma H2AX foci all over it. And we can rescue that with re-expression. So we get gamma H2AX, we get 53BP1, and we get lots of foci. Just so you know that this isn't like, this is specific to cells that have been subjected to 3D migration. So if we have cells living for a week in the absence of FMN2 on a tissue culture plate, first of all, they don't die. And second of all, there's no evidence of DNA damage when they're just sitting around in 2D. And if you irradiate them into, they're on a 2D tissue culture plate and you irradiate them with gamma irradiation, induce all this DNA damage, and then look at the time course of repair, there's no difference in the time course of repair between the knockdowns and the wild type. So this is specific to when the cells are challenged with invasive migration. Okay, it happens in collagen gels too, so it's not just going through a transwell. Cells in 3D collagen gels that have FMN2 knockdown have gamma H2AX foci and 53BP1. And so to sort of add evidence that this is a physical phenomena, we said, what happens if we change the pore sizes of the things that these cells have to crawl through? So we got transwells that had big pores and transwells that had little pores, and we find that there's more H2AX indicative damage as a function of pore size. We can do the same kind of trick with collagen. So if you polymerize collagen at different temperatures, the higher temperature that you polymerize it, the higher the porosity, the lower the porosity of the collagen. And this has been characterized years ago. This is a well-known thing for pure collagen in vitro. So as you decrease the porosity of the matrix that the cell's in, you get an increase in DNA damage in the absence of of this formin that makes this actin structure. Okay, so formins have other things on them besides an actin polymerization domain. They have these other parts that mediate their targeting. Actually, a part of this protein recently has been shown. This protein has recently been shown to have an interaction with P21. So we wanted to make sure that this was due to that the actin, we wanted to know if the actin polymerization activity of this protein was required for protecting the cell from DNA damage and promoting its survival. So the first thing Colleen did was just say, okay, if we perturb the formation of those subnuclear adhesions and actin stress fibers with a drug, is that sufficient to deprotect them? And so what she did is she titrated blebistatin concentrations and found a concentration that got rid of the perinuclear actin and stress fibers underneath the nucleus, but the cells could still migrate through the transwell. So she could still get them to go through the transwell, no problem, but it was like a low dose that sort of just hit the stress fibers, and found that that this enhanced DNA damage in control cells. It had no effect on the DNA damage in the absence of FMN2, but enhanced DNA damage after you stick them through a transwell in control cells. So that sort of suggests that actomyosin is acting in some sort of protective way. And then we made a point mutant. So this was done in collaboration with Pinar, who's in Greg Aleutian's lab. Pinar used to be in Harry Higgs's lab, so she's also a good Formin biochemist, more of a biochemist. Colleen worked a lot on yeast, but based on homology to other known formins, we picked out 2 different amino acids and did point mutations and found this I1226 to A, which has been shown in other formins to kill the nucleation activity, and we characterized that mutant on actin in vitro. And what you can see is as you increase the concentration of the wild-type formin, we did this with isolated FH1, FH2, couldn't get it to work with a full-length protein, but these proteins are known to be auto-inhibited. Nothing's known about the auto-inhibition of this protein, but we had to chop the thing off in order to get it to work, so it's probably auto-inhibited. Anyway, you do this mutation, and you get no nucleation activity. So this is the same as actin alone in terms of the increase in actin polymerization over time. So we got a nice point mutant. So then we do rescue experiments where we knock down the FMN2 and add back the point mutant that can't polymerize actin. It doesn't rescue the subnuclear adhesions. We still see that it's localizing to some little vesicle-y things around the nucleus in 2D. Same thing in 3D. It's localizing around the back of the nucleus. It looks sort of suspiciously unstress fibery in the cell. So we think that it is specifically getting rid of these stress fibers that surround the nucleus, and it can't rescue the DNA damage that's induced by sticking these cells through a transwell. So the polymerization, actin polymerization activity and the formation of these stress fibers is required for protecting the cell against DNA damage when it is undergoing an invasive type of migration. And then to satisfy Andre Nussensweig, he's like, well, what's setting up these there's a lot of biology that I'm not well familiar with about these 2 pathways that induce the repair of the DNA. And we used an ATM and ATR inhibitor and basically found that the assembly of this repair complex onto the double-stranded breaks depends on ATM. So you're probably getting DNA damage here, you're just not assembling the repair complex. This is sort of crazy. It's like actin, first of all, it's actin, it's a nucleator that's making actin that's specifically under the nucleus, that's associated with some adhesions. In 2D, it's sort of holding the nucleus down and keeping the nucleus from jiggling around. So if you get rid of it, the nucleus jiggles around and bumps up and down, and the shape of the nucleus is not well maintained. But if you challenge those cells to invasive migration through confined microenvironments, basically you croak. You get double-stranded DNA breaks. We don't know whether this is due to a physical protection whether this actin stress fiber system is acting like sort of a a flexible force buffer or force, like absorbing some of the force associated with changing the shape of the cell. But my feeling is that it's probably involved in, that membrane-associated thing. That's why I really think that it might be on some kind of extension of the nuclear envelope, and that it's involved in sort of maintaining the integrity of the nuclear envelope. And we're working with Matthew now to know whether this DNA damage is induced by his same pathway where the nuclear envelope's getting torn and the DNases are getting in. Okay, so, it's pretty wild. I mean, I never, you start at the focal adhesion adhesome, and we end up with invasive cell migration and DNA damage. And now we're starting to think more about it in terms of what might, you stop the activity of this protein by killing its actin polymerization activity, and those cells die. It sounds like a good drug target to me. Cells that are specifically crawling through confined microenvironments trying to get out of a tumor. So we're trying to see now whether this protein and its actin polymerization activity are required for metastasis. We're working with Pat Stieg, who's a big breast cancer metastasis investigator at the NCI. We're making CRISPR'd breast cancer and melanoma cell lines where we get rid of the CRISPR, we get rid of the FMN2 and or add back the actin polymerization mutant and assaying for tissue invasion and metastasis. So this project has really taken me places where I've never gone before, and it's been really exciting to work with Colleen and Hawa, who really, she sort of helped us. I mean, Greg actually was the one who thought of, we're sitting there in a lab meeting, actually Greg Aleutian, and he's like, God, that nucleus, we're talking about the nucleus looking messed up, and he's like, I wonder if the DNA's okay. And so that sort of set the whole thing off, and, because we were like ready to try and publish it just about this sort of wimpy phenotype in DNA positioning in nuclear positioning in 2D. And, people have published Nature Cell Biology and Cell papers about actin regulating the position, wimpily regulating the position of the nucleus. So it's not like it's insignificant cell biology, but I was just like, who cares if the nucleus jiggles around a little bit in there, you know what I mean? And if that's all it does, well, we'll publish it, but I want to know whether this thing is important, because this is a really unusual population of actin that nobody's ever paid attention to before. And so, we ran around to Matthew for our microfluidics, to Andre for his expertise in DNA damage, Harry Higgs for his expertise in this particular Formin, and Pat Stieg for her expertise in cancer metastasis. And Colleen has been really the Wonder Woman who's sort of spearheaded this thing, and we're trying to revise it. And she went on the job market before she got this published. She had another paper about a different Formin that was a very nice paper. So she doesn't get a job this year, but I think she probably will. She's like Wonder Woman. I mean, she's like one of the most ridiculously driven and smart people I've ever worked with. So that's saying a lot. She's pretty awesome, and this project wouldn't be where it was without her. She was a national champion in 3 events in karate when she was like 18 years old. So that tells you about her drive and tells you don't want to mess with her. She's really like Wonder Woman. I'm like, if you ever can't make it in science, you get in action flicks, So, if you get a chance to have her come out here, you should do that. So thank you for your attention. Way back.